advancement in the world of liver repair using organoids, producing a new strategy that might reduce the need for liver transplants for like a huge proportion of people who need them, like a third of adults who need liver transplants and like 70% of kids. So today I wanted to talk about a sort of similar study that comes out of the University of Wisconsin in Madison, another fabulous research institution that may yield some pretty game-changing treatments for a completely unrelated disease, Parkinson's disease. So I suspect that Parkinson's is one of those conditions, which most people have likely heard of, right? Perhaps we should just, despite that, we should probably just, sort of do a quick review of what Parkinson's is and how it comes about. So fundamentally, Parkinson's disease is a neurodegenerative disease, right? And it results in a variety of symptoms. Probably the most famous symptoms are the Parkinsonian tremors, right? Which typically look like, fairly rhythmic and importantly, involuntary muscle contractions and relaxations. In other words, shaking. And so I think, the tremors are often like the only symptom that people who've never encountered, the condition think results from Parkinson's disease. But there's a whole variety of symptoms that are also associated with the condition, as well as, importantly, the currently available treatments of the condition, which is a whole other sort of domain of concerns when it comes to Parkinson's disease. So other symptoms include things like having difficulty walking, sometimes appearing like people are shuffling their feet. People will sort of tend to slide their feet along the floor in small steps rather than a more typical stride. And then beyond that, there's a whole set of mood-associated conditions that can arise in Parkinson's, depression, anxiety, a sort of like disconnectedness or apathy can arise with Parkinson's disease. But then even beyond that, broader cognitive impairment and dementia can arise, as the disease progresses, as well as, and I think many folks are unfamiliar with this, substantial changes in sensory systems, like the sense of smell. And there are a bunch of other changes that can occur, like gastrointestinal issues, a flattened affect, where people just like don't really show like emotional facial expressions, and then a variety of other problematic symptoms. And so all this to say, this is a much more complicated condition than I think it's widely appreciated as being. And also, by the way, it's fairly common throughout the U.S. And so here we go. By the way, I think whomever determines the color schemes for CDC's graphics, Centers for Disease Control and Prevention, whoever does their graphics needs to have like a very good talking to, because like various shades of blue and green, it's sort of fairly inscrutable. Maybe I just have bad eyes, to be fair, but I don't think I do. But why pick such like thoroughly similar colors when you're trying to like demonstrate differences between different states? Anyways, not important, not related, but the point is that it's not like there's just one part of the country that faces a disproportionate share of individuals who confront Parkinson's. This is a condition that affects people no matter where they live throughout the United States. And to be clear, this is a map of mortality, okay? Meaning like the rate of people who pass away due to any given condition, in this case, Parkinson's. But, and this will be an image from an article, I believe in The Lancet, which is a medical journal in the UK, a highly regarded medical journal. And so, it's not like this condition is particularly like US centric, right? These are data from 2016, but I imagine the relative trends have largely stayed pretty much the same. And so, anyways, most parts of the world face Parkinson's disease. And I'm sure, there are some interesting explanations for why some parts of the world seem to tend to have like lower rates. And I sort of bet that it doesn't have all that much to do with like pure biology or genetics or anything like that. But, the point is this is a fundamentally human condition. Okay, so I won't get, too far into the brain stuff at this point, but in case you're interested in what part of the brain specifically experiences the progressive, neurodegeneration, the progressive damage that causes Parkinson's. Here we go. So this is a part of the brain, this is the part of the brain on which we're going to focus, right? Or at least part of it. So the substantia nigra. Okay, so as this picture indicates, it's a specific part of the structure called the part of the substantia nigra that is most directly affected in Parkinson's, called the pars compacta. Don't worry about this, but that's what's principally affected. And again, don't worry about the names or anything. It's just in case you're interested. But so this is a part of the brain that has like a whole ton of dopamine producing neurons, a neurotransmitter dopamine. And it's right along the bottom of the brain, sort of near to where like the brainstem kind of like inserts into the sort of bigger part of the brain, the cerebrum basically. And it's what experiences degeneration during the progression of Parkinson's. And so I can't really help myself here. But this is a part of the brain that I explored pretty thoroughly during my PhD research, because it was right next to one of the parts of the brain on which my work focused. Okay. So this is an image that I took a while back. So, don't worry about trying to figure out what you're seeing, right? But I put red ovals, which I hope you're seeing. Yeah, it's a little dim, but I'm putting red ovals around where the substantia nigra is in this brain. And all those little green dots, by the way, are neurons in the brain that produce the inhibitory neurotransmitter GABA. Okay, which, as you can see, these neurotransmitter producing neurons are throughout the substantia nigra. And, I think like a misunderstanding that a lot of people have is that brain regions tend to only produce one neurotransmitter. And that's not the case. So the substantia nigra includes neurons that produce a variety of neurotransmitters, right, including GABA. But it's the dopamine-producing neurons that begin to deteriorate as Parkinson's progresses. Hey, Deanna, good to see you. Hey, Adam from Los Angeles. Awesome. Thanks for joining, man. Yeah, share a cure, really. That would be awesome. Yeah, so we're getting there. We've watched Al go through that. Is that an accessible section? Yeah, I love these photos. Oh, thank you so much. I haven't got to watch you in a while, young man. Thank you, Jim. I'm not so young anymore, I'm sorry to say. Looks like a ram. Yeah, there's a lot of sort of like, not anthropomorphic necessarily, but like parts of the brain that begin to look like faces or things like that, which can be kind of fun, particularly when you're a little delirious at the microscope for several hours straight. But anyways, so this is an image that I took, and those little red ovals, that's where the substantia nigra is. And then probably the thing that's making you think it looks like a ram, those sort of like curly sort of things, that's the hippocampus along the outside. There's the cortex on the very outside. And then just beneath that is the hippocampus, which you might have heard of. So what happens in Parkinson's disease is the dopamine-producing neurons in someone who's developing Parkinson's begin generating a faulty form of a protein, right? So this protein is called alpha-synuclein. And so when that faulty protein, and it's faulty because it's been folded incorrectly, right? So a protein is like basically a string of amino acids, a string of molecules, sort of reasonably large molecules that are all connected together. And then depending upon which molecules you have in which sequence, you get different shapes, right? You can get a dopamine receptor, you can get a muscle fiber, or you can get alpha-synuclein. And all of those things have the opportunity for the protein to be misfolded or folded incorrectly. And that's what's going on in Parkinson's, right? And so when those faulty proteins begin to accumulate, they start to interact with each other and sort of like clump together, right? And so that's what you're seeing in that sort of like brownish red splotch circle in this image. You can think of it sort of like how lint forms in your pockets, right? So fibers from the cloth that form your pockets, it starts to sort of break and fray, and eventually it'll fall to the bottom of your pocket and then start to accumulate with the other fibers that have fallen off and broken, and they begin to form sort of like nuggets, right? Because they're interacting with each other. That's sort of a similar process that's going on with Lewy bodies. So that little red splotch thing, that's a Lewy body. And by the way, this is similar to what goes on in Alzheimer's disease, right? Different proteins, so amyloid beta plaques, amyloid beta, and neurofibrillary tangles from hyperphosphorylated tau, just different proteins, similar sort of problem. They're being misfolded, misprocessed, and then they begin to interact with each other. So, because a specific protein isn't being manufactured by the cell correctly, the errors produce a protein that just doesn't behave properly, right? It's like you can think of it like those proteins have like little magnets stuck on them somewhere where they're not supposed to be. And as the number of those incorrectly produced proteins begins to increase, those magnets will begin to interact with each other and they'll just begin to clump together, right? It's actually quite similar to that process. And since those clumps, those Lewy bodies, as you know, aren't supposed to be there, they can start to mess up what's going on inside of neurons. And as more and more neurons tend to accumulate these clumps, the neurons become less capable of, executing their function, of just doing what they're supposed to do. And then that manifests in all of those various symptoms we were, discussing at the beginning, everything from the Parkinsonian tremors, the motor abnormalities, but then also the cognitive and the mood associated abnormalities. So that was a sort of crash course in Parkinson's disease. We can bid our Lewy body friend farewell. So let's get to today's pretty awesome study. So this is a study that was published in Nature Medicine, which again is one of the most prestigious journals that any scientist anywhere in the world can publish their work. And this paper is by a team at the University of Wisconsin at Madison. Okay, and so this is a preclinical study, which means that it wasn't performed in humans, but it was performed using rhesus macaques, right? So macaques are probably what comes to mind when you think of the word monkey, right? They look like monkeys. and importantly, these are monkeys that exhibited, the ones with which they worked or with whom they worked, exhibited a sort of monkey form of Parkinson's disease. So in other words, this team was trying to cure Parkinson's disease in a group of monkeys, right? You can think of it that way. So it's preclinical. And so fundamentally, the headline of this study is that this team grew neurons from cells that were derived from the monkeys themselves, okay? And then, by making stem cells from the monkeys, they then infused those stem cells. They infused those stem cells into a part of the brain, like millions of these stem cells into a specific part of the brain. Okay, so they took cells from these monkeys, right? Turned them into stem cells, and then basically they turned those stem cells sort of into a primordial type of neuron, and then they infused those neurons into a part of the brain. Okay, so that's pretty awesome. And this is the kind of thing that's been discussed for Parkinson's disease for years and years. And a little bit on that towards the end here that we'll talk about, because it's pretty cool. Hey, Betty, good to see you. Hey, Isa, good to see you. Thank you very much. Hey, Pablo, good to see you, man. Vanita, hello. And hey, Asha, good to see you. Hey. And oh, Michelle, thanks for joining. So this team did a few other things, right? That'll help us to understand the shape of it, what this treatment might be like, right? So first of all, they tried two different kind of strategies, right, for how these cells would be created, and then how they'd be infused into the macaques, right? So the main difference was whether each monkey was infused with cells that were derived from the same monkey, from the self, let's say, or if they were infused with cells that were derived from some other monkey. Right. So in other words, if you had Parkinson's disease, right, would the cells that a physician might infuse into your brain come from your own body, or would they have, they just be cells that they had sort of sitting around and ready to go that aren't derived from your body? Okay. And so we're going to do a little bit of jargon, but don't really worry about the words. We'll just go over them in case you're interested. So the terms for these two strategies, like cells from the self, cells from others, it's allogenic is the term for cells that are derived, or really anything that's derived from somebody else, and then autologous, right? Which is when cells or whatever are derived from your body, okay? And monkeys that received allogenic cells, meaning from another monkey, they got about 5.5 to 6 million total cells infused, and then they infused somewhere between 11 and 22 million cells for the monkeys that received an autologous infusion, right? So cells from themselves. And so I don't really know why there's such a significant difference in the numbers of cells that these two groups received, because it's kind of an important part of their study. And so I'm sure that's something that they had to deal with through peer review, but I didn't see any explanation. So, that might be a meaningful concern. But so let's cut to the chase. So the monkeys that received cells derived from their own body were the ones who improved, okay? And so here is, okay, so this is also a fairly inscrutable image. I know, believe me, just a bunch of color splotches, but just think of it as a sort of heat map of where dopamine related activity is occurring in the part of the brain that's both affected by Parkinson's disease and that they infused with their cells, right? And so on the bottom, that bottom group of 8 pictures, these are the monkeys that received autologous cells, right? Cells that were made from their own bodies. And I don't know if you can see them. Hopefully you can. You can see these little white arrows pointing to a specific color splotch, right? And so those arrows are pointing to areas where in the brain, after these monkeys received those cell infusions, they experienced a rather significant increase in dopaminergic activity. In other words, dopamine producing or dopamine related activity. So, the cells seem to be successfully integrating into the brain. And not only that, but they were also exhibiting the kind of activity that we would want to see, that are intended effect, right, if they're infused into a person with the intention to treat Parkinson's disease. And so, can't really see very much of that recovery in the group that received allogenic cells, right? Cells that were not derived from their body. Those are along the top. So I know it's just like inscrutable color splotches. But if you see on the very bottom row of those images, let's see if I can, nope, wrong side. Okay, so, yeah, you can I'm sure you can sort of see that little white arrow. That is the part of the brain where they infused cells, and they started to see a significant increase in dopaminergic activity. So very cool. And that's only in the autologous receiving monkeys, so the monkeys that receive cells from themselves, or that were derived from themselves. And they also found that the cells that were infused not only survived, which is not a sure thing, but they were putting out some pretty legit fibers, some connections, and seemed to be like legitimately integrating into the brain region. And so they looked like pretty normal neurons. And so here, they have an image here. This is sort of an old school kind of way to do it. But anyway, so, it looks like the cells are doing what we want them to do, right? So you can sort of see, it probably just looks almost like a cotton ball, right? If you were just like spread a cotton ball out flat. All those sort of little black lines, those are neuronal fibers, right? And so that means that the neurons that they injected were actually, integrating. And this, relative to the monkeys that received the allogenic infusion, so cells that weren't derived from their own body, they didn't see these kinds of fibers. So that suggests that there's not only a significant effect of the cells either being derived from the self or from the other, but also that these successfully were integrated. They survived and they integrated. So it looks like the cells are doing what we want them to do. That's great. But only when the cells were derived from a person's own body, right? So what about the thing we actually care about, though, right? That's very important for neuroscientists, of course. But what we actually care about when it comes to this is, did the symptoms of Parkinson's improve? And so within 6 months, the monkeys that received those autologous cells, right, the cells from their own bodies, they started significantly improving, along with about triple the levels of dopamine signaling within a year of the cell infusion. So it takes a while, but that's a huge, huge increase. And so as we noted earlier, Parkinson's is not only associated with movement abnormalities, and they saw improvements in that, the tremors and stuff like that, fine motor control, the ability to just like pick up a cup or pick up a phone or something like that. They saw improvements in that, but also a variety of other symptoms, right, are associated with Parkinson's, many of which can be measured in monkeys, fortunately, particularly mood-related abnormalities, right, like depression and anxiety. And so it turned out that not only did the monkeys exhibit improvements in their ability to control their movement, right, fine motor control, reduce tremors, these kinds of things, but they also showed significant improvements in those other symptoms of the condition as well, mood-associated behavior, things like that. And so, once again, this was only the case for the, the symptom improvement. This was only the case for the monkeys who received autologous cells from their own bodies. And so, and that was not present in the allogenic group, the group of monkeys who received cells from some other monkey. And so this is excellent news, right? For a long time, regenerative medicine, treating this condition with stem cell-based approaches, that always struck me as like a totally intuitive, appropriate, solution to the problems that arise in Parkinson's. And it's not like this has been, this has been an effort that's been pursued now for quite a while, but working with stem cells is extremely tricky. To get them to do what you want them to do, to become what you want them to become, and to successfully integrate. That's pretty tough. And so I noted, hey, what's up, Peter? Good to see you, man. Hello, Vanita. Yeah, oh, wow. What was the myelin sheath activity? So presumably there was plenty of myelination along the axons. They didn't stain for any like myelin markers, but I think the assumption would be that, yeah, there's myelination. But it is a totally reasonable question, and I don't believe that they might have covered that in the supplementary sections that aren't in the main publication. Like you have to sort of click through, and these are just sort of like, oh, and by the way, we also did all this work. And I wouldn't be surprised if somebody asked them to do that through peer review, but I didn't see it in the publication. So it's a good question. So at the beginning, I mentioned a kind of crazy story about like the early days of this kind of a project, this kind of a clinical approach. And so I talked about a story that came out in 2020, a while back, right, about a clandestine procedure that was performed on a physician who had become rather wealthy, right? So he had a lot of money, and he was also diagnosed with Parkinson's. He was shocked to be diagnosed with Parkinson's disease. And so, as they typically do when it comes to Parkinson's, as part of the reason that this type of work is so exciting, the medications that he was prescribed began to stop working. Right. And he decided that he wanted to get a little bit more aggressive with his treatment. And he literally met some scientists at a stem cell conference that, he was not a stem cell scientist. He was not, that was totally outside of his purview, but he went to a stem cell conference. Right. Which is very unusual. It's very, very, I've been to these conferences. It's extremely unusual for someone to just sort of randomly go. Like usually it's somebody in a lab who's doing relevant work, or companies that provide, materials or like microscopes and reagents and stuff like that who attend. Usually there's not very many people who are just like, hey, I'm interested in what you're doing because it might cure my disease. So anyways, a long and interesting story short, I really recommend you seek this story out. He literally funded the research of a highly accomplished scientist, stem cell biologist, who was working, in this area. Specifically in essentially in induced pluripotent stem cells. So stem cells, just like this team did, where they take a cell from some part of your body that's already a skin cell, it's already some other type of cell. They treat them with some alchemy, basically. It's a couple transcription factors and enzymes and stuff like that then basically like convinces the cell to de-differentiate, to sort of go back to a more primitive stage where it's no longer a skin cell, it's a stem cell. Right. So that's what this scientist was working on. And so this physician who was diagnosed with Parkinson's, he met this scientist, and he funded the research that this scientist was doing to the tune of several $1 million, which is pretty unusual. Sometimes like foundations, private institutions will allocate that level of funding. It's pretty unusual for just like a single person who's not like a multi-billionaire to just be like, here's $2, $3, $4, $5 million. Like that is very unusual. But that's the kind of person that this guy is. And so, so the physician, his ultimate goal, very pragmatic guy, the physician wanted to have an experimental procedure performed on him. So he even reached out to a neurosurgeon that he thought would be like the best capable of performing the surgery. And so keep in mind, like basically no one expected, for the people involved, or no one like was aware that this was happening. Right? Nobody even knew that this was going to happen, apart from the people who were like involved, which is like pretty mind blowing. Like in the world of cutting edge biotechnology and medicine, that's usually not how this kind of thing goes. And while things like this have been tried in the past, not identical to what this physician who was diagnosed with Parkinson's received, but sort of similar approaches using regenerative medicine to try and treat Parkinson's disease, they'd been tried in the past, but they were largely, either failures, or they just didn't really produce clear results either way. And, they weren't as stellar as, everybody was hoping. And, we're talking like years and years ago, these types of things were attempted in an era where stem cell biology was still not particularly sophisticated and mature. So now it's significantly more mature. And so there's this whole cool story behind how difficult this was to pull off, like right out of a Michael Crichton novel or something, because the cells had to be produced in a lab in Boston. Okay, Massachusetts, and then flown via private medical jet to Manhattan, to an airport near Manhattan, driven from the airport to the hospital at which the procedure is going to happen. That's through Manhattan traffic, right? And then be infused into someone's brain, like literal brain surgery. And this all had to happen within like just a few hours before the cells would start to deteriorate, right? There's, the clock is ticking. And so it's a pretty awesome story that like I think needs to be made into like a Netflix movie or something like that. But anyways, I suggest you check out this story because it's just fun. So search for something like George Lopez Parkinson's stem cells, right? And George Lopez is the physician, the Parkinson's patient who's also a physician in this story. It's not the comedian, George Lopez. Anyway, so Stat News, I think, by the way, has the best write-up of this story. It's pretty wild. But so all of this happened several years ago. Because, of course, your question might be, well, how's he doing? Did they cure his Parkinson's? Great question. And all this happened several years ago, okay? And I haven't seen much reporting on how Lopez, the Parkinson's patient, is doing, apart from like a few short, sort of positive statements on, like science news sites devoted to Parkinson's disease. And so the evaluations that were done on Dr. Lopez suggested that the cells that they had infused several years ago, I think in 2018 is when they did it, and they infused 4 million cells, by the way. So that's far fewer than the cells that were used in the study I just discussed. But the initial evaluations or the early evaluations of how Dr. Lopez was doing did suggest that the cells did successfully integrate and were indeed producing dopamine. But I recall reading that his improvements weren't particularly groundbreaking, like not stellar necessarily. It seemed more like they pretty much successfully halted the progression, the progression of degeneration, right? More so than reversed it in Dr. Lopez. And frankly, given that I haven't found any more sort of thorough descriptions of how he's doing, it might be safe to assume that he hasn't experienced a miraculous recovery. Don't get me wrong, the initial reporting or the reporting when this story came out suggested he was very, very optimistic, and he was far more sort of optimistic and sanguine about how things were going. He used to surf, and then he went back to surfing. He had to stop surfing when he was being treated for Parkinson's. But just because we haven't heard Dr. Lopez no longer has Parkinson's suggests to me that there is room for improvement. And so in this sort of Jason Bourne style treatment of a human, the scientists were a bit more conservative, with how many cells, literally the number of stem cells essentially that they infused into Dr. Lopez compared to the monkeys in this study. So it could very well be the case that with more cells infused, greater recovery would be possible, totally possible. But so where does this leave us? Okay, so I know that the group in Wisconsin that published the paper that we just discussed is interesting in beginning to explore more human trials as soon as possible, and frankly their results are pretty encouraging. There's always this gulf that separates preclinical research from clinical research, from research with animal models and research with humans. But just the fact that they didn't see any tumor growth, which is always a kind of a concern with stem cells because they are stem cells, they can become anything, or at least they were generated from stem cells, right? So that they didn't develop in the sort of natural milieu that most cells, all cells in your body do, and so they could become other things. And we've seen some pretty gnarly stories of what happens in some folks who get who seek out stem cell treatment that is not FDA approved in the United States for various conditions, some of which report fabulous results, some of which report pretty gnarly and gruesome side effects, off-target effects. I believe former governor of Texas, Rick Perry, actually also former secretary of the Department of Defense or of Energy, I believe he had a stem cell therapy done on his back, if I remember correctly, which was sort of a whoa. Anyways, so just the fact that we didn't see any kind of off-target growth or anything like that, they saw significant improvements in dopamine signals, right? So this is sort of one of the main. Well, this and I guess this one. These are the sort of two main results that I think a neuroscientist would look at and say okay, increased dopamine, neuronal integration, anatomical integration, fabulous. And the fact that they saw, improvements, and I should say significant improvements in fine motor control, the clinical scores that, you can sort of rate how severe a person or an animal's a case of Parkinson's is, it's a number scale. Saw improvements in all of that. And they saw, the rhesus macaques, as they had developed Parkinson's, they would have to grab onto the sides of their habitats to pull themselves up. The macaques that received the autologous cells no longer had to do that, and they had very significant improved fine motor control once again. And they weren't exhibiting, all animals, including humans, exhibit some pretty classic behavioral phenotypes when we're highly anxious or stressed out or depressed, right? And they can be sort of startlingly similar across different species. But so for example, in monkeys, but this is also true in other apes, but then also rodents, dogs, when they're highly stressed, they'll begin to excessively groom themselves and excessively groom other, their sort of peers. And so you'll see like bald spots and like even potentially lesions. They can like lesion their own skin, and you see that also, sort of similar conditions like, trichotillomania in humans. And so you can sort of detect that, given the fact that anxiety and depression are two classic mood-associated symptoms of Parkinson's, and they saw significant improvements in those too. So, I mean, these are all like good signs. Given that it's not like they're the first group to try this. And it's not like they haven't tried this in humans now, right? We do have a clinical trial of one, Dr. Lopez. And so, normally if this was just the first paper that came out, stem cell-based treatment, potential treatment for Parkinson's, normally if somebody were to ask, like, how soon do you think this is going to hit the shelves? How soon am I going to be able to receive this treatment? Or is my loved one going to be able to receive this treatment? I would say, 10, 15 plus years. But, this is an area where there has been some serious progress, and some of which was clandestine, as I said. And so it could be a lot sooner, right? It's not like they have to invent a whole new technology to make this happen, right? It's just injecting cells into a part of the brain. We can do, brain surgeons are good at doing brain surgery. Stem cell biologists can grow stem cells. We know how to induce pluripotent stem cells from your own body, which obviates a whole variety of concerns when it comes to introducing novel biological material into anybody's body. If it comes from you, right, that means that your immune system is significantly less likely to recognize it as foreign and attack. Right, that's like the central concern for any transplant, let alone a stem cell injection from somebody else's cells. And by the way, that is what they believe happened with the monkeys that received the allogenic cells, right? The cells that were derived from some other monkey, not themselves. They believe that it was the immune system that essentially prevented those cells from successfully integrating and, likely attacked those cells. So, I mean, this is really fabulous news for, anybody who has Parkinson's disease, obviously, but also for the people who love them, right? Because Parkinson's is, a devastating condition, and for which we don't have any real, we certainly have no cure. And the treatments that we have their efficacy only lasts for, a short period of time. So that's what I have for you. Sorry, I came in late with the cells infused with the VTA. Yeah, substantia nigra are both. So good to see you, Abyss, our good friend Abyss, always bringing excellent insights. You would think so, right? You would think so. So what Abyss is saying, so the substantia nigra is the brain region that I talked about earlier. Actually, I believe this image shows them both. So it just shows substantia nigra. Don't I have another one that shows? Shoot. So the substantia nigra is the part of the brain, the dopamine producing part of the brain, specifically the pars compacta, that deteriorates. Something like 70% of the neurons in this brain region will begin to deteriorate in Parkinson's disease. And you can see sort of where it is, towards the bottom of the brain. Very old, evolutionarily ancient part of the brain, but it's right next to a more interesting part of the brain that I studied. Actually, you know what? You can see it on my image from my experiments, actually, which is kind of fun. Here, I will make it big so we can just actually see it. So those two circles, those two red circles, that's where the substantia nigra is. You can see part of the pars compacta. In the middle, that little egg-shaped structure with all the little green dots, that's one of the parts of the brain that I studied, the interpeduncular nucleus. And between those two brain regions is the other part of the brain that Abyss brought up called the ventral tegmental area. Okay, and so that is the part of the brain. If you study addiction, if you study the effects of drugs that people take recreationally, that administered to themselves, including caffeine, that's a part of the brain you care the most about. And it is another very, very densely dopamine-producing part of the brain. And they both send their dopamine to similar parts of the brain. However, slight distinction. So the substantia nigra sends, let's see, does this one have it? The substantia, yes, it does. Awesome. Caudate nucleus in the putamen. So the putamen. People pronounce that differently. But anyways, substantia nigra and the ventral tegmental area send their dopamine to a part of the brain towards the front, as you can see, called the striatum or the striatum. People pronounce it differently. But the substantia nigra tends to send most of its dopamine to the dorsal striatum, so the part of the striatum, again, towards the front of the brain, towards the top of the head, okay? Whereas the ventral tegmental area sends most of its dopamine to the ventral striatum, a part of the brain called the nucleus accumbens. And so, you can just think, you divide like my hand in half, right? Ventral tegmental area is dopaminating this part, and the substantia nigra is up here. And so Abyss, they infuse these cells into the caudate and putamen, into the dorsal striatum. Kind of weird, right? But I guess not, right? Because I don't know if you ever saw the story of this, of the story of Dr. George Lopez, not the comedian, who underwent an experimental procedure where he was injected with essentially stem cells. I think for him it was neuroprogenitor cells. They injected into the caudate and putamen as well. So I don't know if there's my initial thought, and I know this is getting like a little bit nitty gritty for probably most people, but just bear with me for a second. Like those projections from those axons, those cables that the ventral tegmental area and substantia nigra send to the dorsal striatum, or to the striatum in general, those are pretty long projections, like really rather long, among the longer projections in the brain, literal like physical cables, axons, right? And so I can imagine that it may be almost impossible without, recurring surgeries to get those axons to grow, right? To successfully integrate into the, mesostriatal circuit, to the striatum, all the way to the striatum. I could imagine you could inject some chemotactic, agents, some hormones that guides the growth of those axons. But at a certain point, it's like, you're in brain surgery like every other week. And so I could imagine that just having dopamine producing neurons right there, maybe that's all you need. I don't know. I could also just be completely misunderstanding what they did. It's possible, but that's sort of what came to me, is that like it's much easier for a neuron to send an axon to a neuron that's right next to it, essentially, or nearby at least, than it is from the posterior part of the brain to the anterior. Can you please say hello to? I don't know how to say that word. I'm sorry to say. Augie, maybe is your name. Hello. Oh, hello. Yes, thank you, Vanita. Okay, I think Ian is from the medical future. How could you know this much stuff? I'm waiting for his book. I would love to write a book. I have so many ideas for books, but I don't know how people actually do that. But that's very sweet of you to say, Kent, but it's my job. There are things about which I know very little. Most things, the vast majority of things. And there's far more in neuroscience that I don't know than I do. But I appreciate the sentiment. Thank you very much, Sherry. I appreciate the award, and thank you, Peter. I really appreciate that. I've seen that in a client that had Parkinson's when I was a caregiver. Their lack of mobility just keeps increasing along with the anxiety. It's a totally changed quality of life. It would be like a fundamental game changer. Like all the medications that we have for Parkinson's, they're glad that we have them, but essentially they all revolve around essentially like artificially amping up that dopamine level using a combination of carbidopa, L-dopa, basically like just putting more dopamine in the brain, kind of, or implanting electrodes and basically just like injecting current into, specific parts of the brain. And, you literally have like an electrode implanted into your brain, and it's always there. And there are some really, I was considering snagging some videos. There are some amazing videos of Parkinson's patients who have pretty advanced severe Parkinson's, and they have these stimulators, and you can see what happens to their tremors when they turn off those electrodes. The tremors, almost within maybe 2 seconds, the tremors. Somebody who had been completely, you would never assume has Parkinson's disease at all or has any kind of movement abnormality, they begin like very overtly, powerfully, shaking. And I've seen there's quite a few on YouTube. You should really check them out. I've seen one where the tremors became so severe that the gentleman was having difficulty turning his stimulator back on. Like that's how severe we're talking. And then, when they successfully turn it back on, within seconds, gone, right? I think it's one of the most powerful demonstrations of the relationship between brain activity, behavior, consciousness, where it's just in 1 second, the only thing that changed was a little bit more electrical current. That's it. And then all of a sudden, this person is completely different within seconds, right? So I recommend you check them out. They can be a little, I've shown them in to a class, an intro neuroscience class, and it can be like a little jarring, I think, for some people to see, particularly if they had a Parkinson's patient in their, or somebody diagnosed with Parkinson's in their family. Your graphics are better than CDC. I appreciate that. Hey, Cosmic Rose, I appreciate you joining. Is there a link to your thesis? There is, Rob. Yes, my thesis is publicly available, but I would not inflict my thesis, my dissertation on anybody. I didn't even ask. I urged my parents to not read my dissertation because it is just dense. And it's also several hundred pages. So, I mean, but hey, read at your own risk. Yeah, I mean, you should be able to just search like Ian McLaughlin. I should put a link on my website, but you should be able to just write like Ian McLaughlin, PhD, University of Pennsylvania, IPN, and you should be able to find it because it's on a public repository. You know, at many programs, if not most, it's like a requirement that you, unless there's any kind of like any kind of intellectual property, that you make your dissertation publicly available. And it's important. It's important because a lot of rising scientists, me, included, would end up referencing or at least looking to other dissertations. Just because science wasn't necessarily published in an academic journal doesn't mean it's not potentially insightful. So yeah. Thanks, Kent. That's very sweet of you. Which reminds me, do you think the efficiency of Parkinson's treatment can be enhanced if, say, DBS was administered along with L-DOPA? I've wondered that a bit, if there's a sequence, because I would imagine that you start with L-DOPA, right? And so I sort of hinted at this in the beginning. There are a bunch of side effects of, and I know you're well aware of this, Abyss, but like there are a bunch of side effects of that some people experience from Parkinson's medications. I mean, like really rather disturbing ones. Like, there can be, excessive, like somebody who has never gambled in their life, no real, compulsive behaviors before, no addictions. They are diagnosed with Parkinson's. They are treated with Parkinson's medications, and then they become like their desire to gamble is insatiable, and they end up gambling away tons and tons of cash. They just can't control it. But there are other, arguably more disturbing ones where people become like just their libido goes off the scale, and they're just like, it becomes sort of a problem in their personal life where, they just cannot control themselves. And there's even more disturbing ones where, sexually abusive behaviors can emerge. This is not the norm, to be clear, but these are side effects that are known of Parkinson's, particularly like engaging in riskier behavior, gambling, things like that, excessive drug use. And so, but anyways, I would imagine that regardless, you start with those types of medications and then you escalate to DBS. But once DBS, deep brain stimulation is what Abyss is referencing, the implantation of electrodes targeting specific parts of the brain, injecting literal current. Once that stops working, you're pretty much at the end of the road. And so, but so I would imagine that's the sequence. I'm not entirely sure, and it might be the case where some people have such aggressive Parkinson's that they just start. Let's not even waste time. I would imagine sometimes it's both. It is combined. Hey, T-Bones, good to see you. Oh yeah, for sure. They also have a distorted perception of time and excess L-DOPA, which I find very interesting. Yeah, I think we've even talked about that, haven't we? I believe. All right, I should probably get back to work. Thanks everybody for joining. If you're seeing me somewhere else, I'm using a platform called haps.tv, HAPPS.tv. If you're watching on Periscope, I have a link to it in my profile page. And come and join. Everybody's over at Haps now. No, but seriously, there are a lot of really cool, it's a really cool community at Haps and Haps.tv. And, probably most of the scientists with which you're familiar are there. Deanna C. Hooper, dark matter scientist, coolest way, the closest you can get to like being able to say I'm a wizard. Cosmologist, physicist, theoretical physicist, DC Hooper 91, check her out. Christian Zasa, one of my favorite, easily in the top 10 nature photographers and astrophotographers I've ever met. I've only ever met one though. No, Christian Zasa is incredibly talented. The types of photography and astrophotography, nature photography of like hummingbirds, of eagles, of nebulae, of eclipses, of all kinds of really amazing stuff that you would see in like National Geographics, and in one case literally in National Geographics because they featured one of his pictures of the Milky Way galaxy. You can find him at Zasa, S-A-S-S-E photo, SAS photo, or just type in Christian Zasa. That's how you pronounce it, but it's S-A-S-S-E. Who am I forgetting? I'm forgetting somebody. Oh, we're going to try and convince. We're going to try and convince Abyss to start joining us. Gosh, Issa Betancourt. Oh my gosh, she was the one that invited me to Hats. Sorry. So Isabel Betancourt, or Issa, ISA Betancourt. You can find her. She is an entomologist. She's brilliant. She has access to amazing collections. And if you've ever wondered anything about a bug, she either knows the answer or will get you the answer. And she'll also try and convince you to hold a scorpion that is like absurdly humorously too big to be held by any human, any rational human, which she did to me. So you can find her at Isa Beta Bug, ISA Beta Bug. And hopefully the science community will continue to grow. But anyways, haps.tv, come check it out. I will see you hopefully on Sunday. I might have to miss it because it's my wife's birthday. So I might see you on Monday. But we shall see. Otherwise, have a great weekend. And until next time, stay safe, wear your masks. Let's all get vaccinated. See you next time.